Hi everyone, I'm Sandra Clark, and I want to welcome you and thank you for attending our first talk for the FY 2021 series of the NOAA Environmental Leadership Seminar Series. The goal of these seminars is to showcase NOAA's leadership in environmental science by those who lead it and make it happen. I'd like to take a moment for a few housekeeping details. First, I would like to thank the NOAA Research Council and the awesome team that I work with in scheduling these seminars. Without these seminars could not happen. Hernan Garcia with the NESDIS National Centers for Environmental Information, Tracy Gill with the NOS National Centers for Coastal Ocean Science, and Katie Rowling with the Office of Ocean and Atmospheric Research and the support from the NOAA Central Library. We also want to thank Cal Bliss with the NOAA Office of the Undersecretary and Kate Brogan on details to the Office of the Undersecretary from NOAA Communications for moderating the questions today. Thank you, ladies, for being here. Second, some seminar logistics, most of which are listed in the Q&A pod. Please type all questions and comments into the Q&A box, and we will try to get to as many questions as we can at the end of the seminar. We are recording this seminar for later viewing, and the recording and PDF of the slides will be made available in a few days at the link provided in the chat box. If your system seems to be lagging, please turn off any extra apps you might have on. Also, it would be best to watch this webinar in full screen. Now, looking ahead to finishing out the calendar year for 2020, we have some awesome speakers. Vanessa Griffin with NOAA NESDIS will be our next speaker on November the 10th, and Admiral Gallaudet will be returning to our series on December the 8th. You can visit the NOAA Environmental Leadership Series webpage, which is listed in the Q&A pod. We are hard at work scheduling the rest of our speakers for FY 2021. Today's seminar title is From Orchids to Oceans, Environmental Citizen Science Leadership in Action, and our speaker is Dr. Neil Jacobs, Assistant Secretary of Commerce for Environmental Observation and Prediction, performing the duties of Undersecretary of Commerce for Oceans and Atmosphere. Dr. Jacobs is responsible for the strategic direction and oversight of over $5.54 billion, yes, that's with a B, in annual spending, including key investments in developing a community model framework to advance U.S. weather modeling and prediction, science innovation, streamlining unmanned systems research to provide critical data across NOAA's missions areas, and unlocking the partnership potential of non-governmental and private organizations to study our nation's oceans and promote a blue economy. Previously, as the Chief Atmospheric Scientist at Panasonic Avionics Corporation, he directed the research and development of both the Aviation Weather Observing Platform and Weather Forecast Model programs. He was previously the chair of the American Meteorological Society's Forecast Improvement Group and also served on the World Meteorological Organization's Aircraft-Based Observing Systems Expert Team. Dr. Jacobs holds a bachelor's degree in mathematics and physics from the University of South Carolina and master's and doctoral degrees in atmospheric science from North Carolina State University. Welcome, Dr. Jacobs. Thank you for your leadership and for presenting at the NOAA Environmental Leadership Seminar Series today. The floor is yours.
Thank you. How's my audio?
You sound great.
Perfect. Well, thanks everyone for joining this today, and thank you very much for that introduction and to the entire team for putting this together. I'm actually very excited to be speaking about something that has nothing to do with a lot of my day job, but it's something that I'm very passionate about, and it's definitely citizen science. So I'm just going to dive right in. We will be looking at questions as we go through this, but probably we'll try to answer most of them at the end. So if you've heard anything about me and my time at NOAA, it's mostly probably been with respect to numerical weather prediction and forecast models, things of that sort. That's pretty much my area of expertise. I really spent a lot of time studying East Coast extratropical cyclogenesis, primarily with respect to the Gulf Stream's influence on baroclinic instability. And why would I want to do that? Well, primarily because my main passion in life, when I'm not hunting orchids or fishing, is surfing. And I always wanted to know when the waves are going to be good. And figuring out how to forecast them was essentially just a problem of applied math. So I started getting involved in writing weather model code, which was largely self-serving, so I just know what days in advance to take off so I could call in sick to work and go surf. Here's one of my friends from Surfline, who is now the meteorologist in charge at the Wilmington Weather Forecast Office, Mark Willis. Whenever I do get a chance to go surf, that's probably nearest spot to home, next to the Outer Banks. What I've been working on since I've been at NOAA, among a million other things, has really been focusing on the unified forecast system and the community model. And this actually is citizen science too. What we really wanted to do was get the weather model code out into the public domain so that we could essentially crowdsource model development. Historically, the external community never had access to our code, and if they did have access to the code, if they didn't have login credentials to our machines, there was no hope of them actually running the code. So a lot of really smart, talented folks spent the last couple of years porting the code over to architectures that the community has access to, including actually, believe it or not, running the global model on laptops. And now that the code's out there on GitHub, it's essentially runnable in a lot of cloud service providers' platforms. So the idea is now university students, PhD students, postdocs, PIs can actually download, run the code, do development work. Their motivation is to write papers. My motivation is to get their contributions integrated back into our system. A little bit other ideas on citizen science. So I don't know if folks know this, but my uncle, George Jacobs, just recently received the award. He passed away in 2017, but received the award for 50 years straight of being a cooperative observer in Tok, Alaska. And it was actually kind of funny. We got this certificate. There's my aunt down there actually getting it, and it was signed by Louis Uccellini, and she emails me, and she wanted to know if I knew who he was. But this is the kind of citizen science that folks in the Weather Service really rely on a day in and day out basis, just for analysis, forecast, as well as verification. But it's not just on the weather side, it's also on the fishery side. So there's a couple different fish there. At the bottom, there's a tremendous amount of work. So a big shout out to National Fish and Wildlife folks for doing a tremendous amount of restoration of the brook trout population. This was a native species, but it's easily outcompeted by other more aggressive species. And then on the top left, there's actually a peacock bass that I caught in a canal not far from the National Hurricane Center, which was actually a species that was introduced into Florida. Now, I think folks probably know that we're a regulatory agency on the marine fisheries side, but this also is a component of citizen science, where there's an interesting app. I happen to have one for the southeast states, where you can actually download the app and record information about the fish you catch. And part of being a regulatory agency on the fisheries side is we have to determine what the quotas are and the catch limits, and actually understanding and having a really good idea of how many fish are out there helps us determine where to set those quotas. What I'm going to talk about today, though, is something that actually really, I guess maybe it indirectly involves some work at NOAA, but this is something that I've been doing with my wife since the mid-2000s. I think we started this around 2004, and the idea was, let's just see if we can catalog all the native orchid species. And we started in the Carolinas and ended up deciding we were going to broaden it to the U.S., North America, and update a text by lure. And you can see here there was 2 texts. So he passed away last year, but these 2 texts are the current state of the science, basically. They're almost, well, 1972, 1975. They're quite old. And what's interesting about these is one of them is native orchids of the United States and Canada, and you can see there it says excluding Florida, and the other book of equal size is just the native orchids of Florida. It gives you an idea of how many orchids are actually in Florida. But what we really wanted to do was go and see how are these orchids doing? Are they still there? Now, orchids are extremely adaptable, so things like climate change, they can evolve and adapt to really quick. But one thing that they can't adapt to is habitat destruction. And so knowing whether these populations still exist is sort of a big unknown. So we set out on our quest, and I'm just going to take you through sort of I guess, a little bit of adventure of what we were doing. So here you see Jen, my wife, actually taking a photo of this corallarizer Wisteriana. I'm holding the flash there. And Howard Bridgman, a naturalist down in South Carolina, came along with us to observe. This photo was taken in Francis Marion National Forest. So you can actually see right here the plants. They're very tiny. They're not green. They actually have a relationship with a fungus in the soil that also has a relationship with certain types of trees, and that's how they generate food for themselves. So some of these orchids don't even photosynthesize. So you can imagine walking through the forest floor trying to find something that tiny, which is not like your typical grocery store orchid, is not easy. This is actually a zoomed in view of this. I wish I actually had sort of a scale on here, but this flower is probably a couple millimeters across. These can be found throughout the Carolina coast. In fact, I've seen them at the Whorl Bay National Estuary and Research Reserve as well. So how much do we know about native orchids? Well, the short answer is not very much. And you can see that there we've studied about 880 species, and you can get an idea of how many other species are out there. So these orchids are probably one of the most cosmopolitan plants there are. And you can see there the breakdown of the ones that we have studied, which are not threatened, endangered, critically endangered. One unfortunately extinct, and then some that we have been studying, but there's insufficient data on them yet. So you can imagine that there's a tremendous amount of uncertainty with all these that we don't know about. But just these that we've studied, this number here, the endangered and critically endangered, are growing. But we don't actually know at what rate. And the reason why is they're not being tracked. They're not being followed. A lot of people actually don't realize they're seeing orchids when they see them, so they're not being reported. So the idea behind this project was to essentially inform the public as much as we could and get information out there so that people could report it through apps. And then you're essentially crowdsourcing eyeballs looking at the forest floor, for lack of a better way to describe it. So there's a big program at the Smithsonian Environmental Research Center. And you can see here that if you go to their website, there's citizen science. You can click on that link, and it talks about orchids in the classroom. And they really sort of explain orchids, the plant, the flower, a lot of times the relationships they have with an associated fungus. That's associated with all sorts of other, usually trees, but this is one reason why a lot of these native orchids, the biggest problem that we see other than habitat destruction is poaching. And people will find an orchid, they think the flower is pretty, they dig it up, they take it home, and then it dies. And the reason why it dies is they have a relationship with a typical type of fungus in the ground, and that fungus has a relationship with the tree. And so unless you plan on digging up the entire forest floor, you're not going to be able to sustain this plant. And these orchids actually, each species has a unique fungus that it's usually paired to. So doing a lot of gene sequencing and trying to identify the fungus has been one of the research areas of this particular project, because if you're ever going to propagate these in captivity, you have to understand what fungus the plant's associated with and what other host that fungus needs to exist. Over here on the left, we have what's commonly known as the ghost orchid. The genus is Dendrophylax. There's actually 2 species in this genus. I've been able to, at home, grow the Dendrophylax spinalis, which is the other species in this genus. You may not have heard of this particular plant, but this is the flower/orchid plant that the book The Orchid Thief was written about. So here we have some photos, quite old photos of some power line cuts in eastern North Carolina, where you can see just fields of Platanthera nivea. This is something that we haven't seen in North Carolina for years. Apologies if you can hear that siren. There's a couple of reasons why a lot of these plants are no longer found. Some of them have to do with excess fertilizer. Some of them have to do with herbicides. These particular plants are extremely sensitive to those type of components in the soil. Now, we were actually able to locate this plant, not North Carolina. This photo was taken on the side of a road down near Brunswick, Georgia, but you can see for perspective that there's the plant right there. It grows up on kind of a tall stalk. It's fairly large. It's a couple feet tall, and then you have this flower stalk here. There's a couple more back here. Here's a nice large one. But really, to get a true appreciation of these types of plants, you really need macro photography, pretty sophisticated camera equipment. And this particular plant that Jen was taking a photo of, this is the actual photo. And you can see that this is for perspective. This is 2 millimeters across, so it's actually very, very small. If you were to see this up close, you would think, oh, that kind of looks like an orchid flower. But if you see this driving by on the side of the highway, it just looks like kind of a tall white spike. It's not really, doesn't really stand out. There are some what I would call more exotic, flashy type orchids. The Calipogon genus is one of them, and the Calipogons are all over the east coast of the U.S. The Calipogon tuberosus, actually, I've seen that all the way up into Newfoundland and Nova Scotia. We've got the Calipogon tuberosus down here. This was taken in Cranberry Glades in West Virginia, and this particular, the alba version of this, which is just the white version, was in Brunswick County, North Carolina. The middle one was in Francis Marion, but there's a couple other species in this genus. One of them is pallidus. That's a little bit smaller, and then one is barbatus. These are both more seen in the pine savannas in the Carolinas. Now, there's one particular species in this genus that had not been seen for decades in the Carolinas, and I was fortunate enough to actually be on a business trip down to Avon Park in Florida. I was testing out some UAVs for my actual real day job down there near MacDill Air Force Base, and decided to do a trail run through this reserve and found a bunch of fields that had just recently been burned. And sure enough, there was signs that there might be the Calipogon multiflorus coming up. Now, you can see in the background of the photo here on the left the burn. So there's some palm fronds there, but this particular species of Calipogon is burn dependent. It won't bloom unless there's an actual burn, because it doesn't like competition on the floor of the. It's not really in a forest. It's more in sort of a prairie. And it also, a lot of these plants, the flowers, they're short-lived they all bloom sort of at the same time, and it's advantageous to the plant to synchronize their blooming because they want to have the max exposure to potential pollinators. So if the flowers are short-lived and they bloom at different times, the odds of a pollinator hitting multiple plants are reduced. So one of the ways that they can synchronize their bloom time is trigger it to all happen at the same time after a burn. Now this area in Florida, which is kind of in the central part of the state, would often have natural burns just due to lightning strikes. And because of a lot of forest conservation management and burn prevention programs, it had almost been eradicated further north because of things like fire breaks and other potentially, let's say you do controlled burns, which in general are great, but if you do the controlled burn in the winter or when it's not in the normal season that this plant would be expecting to bloom, you're still going to mess with the plant's ability to actually reproduce. So there's been a lot of research done at this particular preserve about actually doing prescribed burns during the proper season when they would expect to happen just due to spring thunderstorms and things like that. And sure enough, that this plant down there is flourishing. So this the Superpedium is probably a genus of native orchid that a lot of people are familiar with. I'm sure if you've probably been out in the woods and seen a native orchid, you might have seen the pink lady slipper, which is a Superpedium Helle. This is a Superpedium kentuckiensis. And what's interesting about this particular plant, besides it's actually quite large, and this photo was actually taken on the day of the Kentucky Derby, was that this is a disjunct population. I'm excited to actually say it is within walking distance from Mallows Bay, over on the east side of on the eastern part of the state. It's typically otherwise found in the mountains, in the mountains of the Appalachians, and then I think even west. But it's very odd to have this down by the coast. But it's doing quite well. Here you can see it's you can get perspective on how large it is. It's quite it's I believe it's the largest of the lady slippers. And we actually walked through quite a significant amount of mud before we were able to find this. But this is part of what's exciting. A lot of times, if you're just hiking through the woods and you're on the trails, you're probably not going to see orchids unless they're very, very nondescript, tiny green ones, which no one else would recognize. That's unfortunately because usually the flowery ones alongside the trails have been dug up. So typically, if you want to go orchid hunting, the best thing to do is get off the trail. Of course, bring a compass. I've got an interesting story about looking for orchids in the woods adjacent to Quantico, and I actually think something was going on with the electronic compass in my phone, which wasn't working properly for some strange reason, and ended up having to use a magnetic compass to find my way out. That and the traffic on I-95, which was an indicator of which direction I should go. But I have been lost in the woods. Not particularly fun, but if you want to find interesting stuff that no one's seen before, that's the only way to do it. So a couple close-up photos of this, which I thought were kind of cool. So this is actually Jen taking the photo and me playing around with the strobe. These windows, these panes that you see in the back of the pouch, are actually there for a reason. So a lot of times what will happen, and I think it's a misunderstanding, these aren't carnivorous plants like pitcher plants or things, but they do have this pouch. And bugs will go down in the pouch. They won't get stuck there, but they'll head towards the light. And they head towards the light because they can see the light shining through these membranes. And what happens is they head towards that light to get out. And when they go out, they end up going through here, and they cross this zone and end up getting pollen on themselves. So it's kind of a gimmick from the orchid to attract a bug to crawl up this way and come out and get pollen on itself. It actually helps it fertilize other plants. So here we have Liparus lowfolia. This was a photo taken literally right off the side of the trail in the Eno River in Durham, North Carolina. And I thought this plant was actually pretty cool. One, because it's really, really small, but also because the lip, if you hit it with the light just right, it's translucent, which is, I thought, really cool. But in order to get a photo like this, where it was just tack sharp, and hopefully on your screens it shows up pretty crisp, you need it to be extremely still. So Jen was taking these. I can't remember what her lens was. I think it was like 150 millimeter macro lens. And she kept accusing me of breathing on the plant while I was holding the flash there, because it kept moving around, and I was holding my breath, and I promised her I wasn't breathing on the plant, but it kept moving. And it turns out that there was this tiny little jumping spider just sitting on the edge of the flower, just moving around and shaking the plant the whole time. And I don't know much about arachnids, so I can't really tell you what type of spider this is, other than the fact that it was a really excited one that liked to jump around a lot. So here we have probably the rarest orchid in North America, Isotria medioides. And this, you can Google this. You can Google the common name, which is small world begonia, Whorl. This there's, I think, in the community, in sort of the botany community, there's a lot of thinking that maybe this is a little bit more common than it's listed. So it's listed, and I'll show you. It's on a following slide. It's either threatened, or critically imperiled. But there's a couple issues. First of all, this is literally a very tiny plant. It's maybe 2-3 inches tall. It's green, and it can go dormant for over 10 years. And we only know it's gone dormant for 10 years because we've been collecting 10 years worth of data. It could very well go dormant for longer than that. We just don't know because we haven't been monitoring it that long. But it can go below ground and it won't come up, and it actually lives because it has a relationship with a fungus underground, so that it can actually generate food for itself without having to come up. And that's how it ends up staying dormant for so long. So you can imagine if you go hunting for these things and they're dormant, sorry about that, they're dormant that long, it makes them actually really, really hard to find. And so you may think, well, this particular population of plants is gone, it's not coming back, or who knows, surprise, maybe it comes back threefold in 10 years from now. This particular photo was taken in Prince William National Forest. There's a couple of healthy populations there. There's actually Western North Carolina, and then up in Vermont and New Hampshire, they're doing fairly well for an endangered species, basically. For the photography fans out there, this plant is the exact same plant. The camera was not moved at all. If you actually look very closely at these two photos, you can tell that they were literally taken a couple of seconds apart. And it was just me playing around with the flash while Jen was using the trigger for the camera. So here, one of them is backlit, the other one has two different strobes from the front. So one of the things that we were doing for the update to this lure text was to have really cool, sort of interesting photos that would get the reader interested. So there's plenty of field guides out there. No one really thought we needed another field guide. But what we thought was really cool about the lure text was it's kind of doubles as a coffee table book, and having interesting photos was one good way to sort of generate additional interest in the actual orchid species. So here's a map of the distribution, and you can see these little hexagons here. Not all of them are filled in, because I can tell you there's some pretty good populations over Northern Virginia. But in general, it's a struggling species, and it's also tricky to find. So I just got this clip from this article. So there is a species, Indian cucumber root, Mediola virginiana, that looks almost exactly like Medioloides, particularly if it's a non-flowering version. So being able to distinguish them in the field is tricky. So I'll give you a little quiz here. So here is the only other species in this genus in North America, verticillata. And here we have a picture that I took looking down on both a Isotria verticillata as well as the Mediola virginiana, which isn't even an orchid at all. It's a completely different type of plant. But you can tell they look almost exactly the same. It's the one on the top that's the actual orchid. And you can imagine if you're hunting for this particular orchid and you're walking through the woods and this particular plant is extremely common, it's probably as common as poison ivy, that you're constantly getting your eyes fooled by thinking, there it is, I found it. Nope, it's not. And you actually have to look at the stem to really tell the difference. So here's another orchid that if you saw it, you probably, A, wouldn't think it was an orchid, and B, I'd be surprised if you saw it because it is actually incredibly small. Here's how big a millimeter is. You really have to be looking for this if you want to find it. But Molasses Unifolia, and it's unifolia because it has one leaf. And this particular species is relatively common in the region of the Carolinas up into Virginia. Certainly, this is a top-down view of the exact same plant here. Where it gets tricky to identify is the different species in this, Molasses bayardii. This species has only ever been reported in one location, of which I was fortunate enough to go there with Jen and photograph it. It's at a place just north of, hopefully I'm saying this right, Wacoit Bay. It's another National Estuary and Research Reserve. The zone at the north end of the reserve, halfway between there and a place called Peters Pond, is the only known population of these plants. And there was some big discussion among the botany community of whether this was actually a legitimately different species, or was it just Molasses Unifolia with some different morphological features because of it was the particular components of the habitat there. So it's actually kind of easy if you go in with a high-powered macro lens and take a picture of the actual flowers between the two, and then you can compare them. So if you're going to compare the actual physical differences, it's basically just a ratio of the size of the basal lobe, the labellum there, to the length right here. So if the ratio is greater than 0.6, it's bayardii. If it's less than 0.6, it's unifolia. Now this, to me, seemed kind of questionable, but they've since taken tissue samples and run the genetics and determined, yes, it is different. But there's a lot of this going on in the orchid community, in the native orchid community. I'm sure it goes on in horticulture as well. Lots of hybridization. So here we have several plants. These are actual just common species: Platanthera grandiflora, Lacera, Psychotes. And these are the hybrids. So Keanii is a hybrid. So here's Keanii here, and then Andrewsii is another hybrid. And of course, there can be different shades of the hybrid here. Lacera is typically a green, very green flower. I don't have a photo of it here. The question was Schreiberii a hybrid or a distinct species? And the reason why it got the name of a distinct species versus a hybrid is because it was blooming literally very close and at the same time as another hybrid, but it wasn't the hybrid. So it could have been hybridized a long time ago and then been its own species so long that the bloom times actually diverged enough so that at some point in the recent future there was not really overlap between the two periods of blooming. But you'll notice on this particular image, these types of Platantheras all have fringing. But there is a purple Platanthera called, the common name is Fringulus, which does not have fringe. The Latin name is Platanthera paramonia. And it's relatively common in the mountains of Virginia. By common, if you really know where to look, you can find it. But there was actually a historical record of a population in Durham County at a place called Dial Creek. And I found it going through some archived papers. And I actually contacted the wife or widow of the man who originally found it. I think she was probably in her 80s. And she basically was able to tell me pretty close to where it was. And you would think that long ago that the odds are there would be a strip mall or a neighborhood or something where this was now, but I decided to go look anyway. And it turned out to be kind of a strange adventure. I had to wander through probably 3/4 of a mile of 10-foot tall blackberry bushes, and ended up coming out in the swampy area where you can actually see one there. I think I counted 6 or 7 plants at the time. They normally bloom in early to mid-August in western North Carolina, so assuming that the central part of the state would probably be a little bit ahead of that, I went out in July. And there had been a lot of upstream development, and the upstream development had really slowed down the water flow. So there had been a lot of diversion of the water downstream just due to the development. And you would have expected that to eradicate this particular plant, which likes to almost grow in standing water. And I kid you not, the only reason why this population was still in existence was because a beaver network of dams had completely built around this whole area, retaining all the water. And I haven't been back to actually check to see if these beavers are still there and how the plant population's doing. It's probably been 7 or 8 years since I've been to this site, but these are all the kind of weird, bizarre inner workings of the ecology that are dependent on each other for these things to end up being successful. And here you can actually see a close-up version. I guess this is a fly or some type of insect here, and you can actually see the pollinia stuck to its eyeball, which I thought was kind of entertaining because I guess it's going to go fly to another flower here in a second. So as I said, orchids, most people would consider them probably the most cosmopolitan plant species, and here we have Epipactus Helle, which I actually found. I went outside of my apartment in Arlington one day to go for a morning run. I was running down the sidewalk, and I looked to the side in the ivy and saw this growing there. I thought it was kind of odd, but it was literally growing right by this manhole cover. I don't know if I'd call it an invasive species. It's a native orchid, but it's native to Europe. It's not native to North America, but it's commonly known as a hitchhiker. I don't necessarily think it actually has to have any specific relationship with a particular fungus to be successful, because it is so successful almost anywhere. But if you're ever in the Arlington, Crystal City area in the spring, if you just keep your eyes peeled, you can see this growing all over the place. And a lot of people may not actually realize it's an orchid, but it is. So here's an interesting species, Trianaphora. This is incredibly hard to catch blooming. These flowers last for about an hour and a half each year. And they synchronize their bloom based on weather conditions. So this particular plant was photographed in the Pisgah National Forest just outside of Brevard. And here are, this data is from, I guess, probably the ASOS station at the Asheville airport, which might be the closest, but there's actually some personal weather stations in the area too. So maybe a little bit of citizen science played into this too, but there was really trying to figure out when exactly this was going to bloom. These things will come out of the ground and stay in bud in July, and they will stay in bud for weeks, but they won't bloom until there's about a 10-degree drop in evening lows. So here you can see a cold front comes through, and there was a pretty significant drop between the 7th and the 9th in the temperature, and this plant will bloom. All the flowers will open up at sunrise on the second day following this temperature drop at night. So go there in the morning, 48 hours after the first cold front comes through, and you will see a mass blooming of these things on the forest floor if you're in an area where they're located. But if you're there by probably 10 in the morning, all of the flowers will be closed up, and 2 days later you can see that they're all going to seed. The reason why they do this is just to synchronize the idea that all the flowers are open at the same time to get the maximum cross exposure for their particular pollinators, which I don't know what else is pollinating them, but when I was there, these little iridescent green bees seem to be frequenting a lot of these flowers. Here's another Plutanthera. This also does not have fringe. There's some debate in the native orchid community about the genus, but I am not a splitter myself. So I'll continue to call this Plutanthera. This species is Integra. And again, much like the purple fringed orchids, this one also has quite a few hybrids. So here at the top we have cristata, ciliaris, and Bepharaglottis, which is the white one, and then we have the hybrids, Chinellii and Laurii. And Laurii, I think, is probably the most obvious hybrid between the white one and the orange fringe, because it basically looks just like them, except the color's halfway between. These are all the different things that go into forming the database that you would look up and report these plants when you see them in the wild. And knowing exactly which ones you see and being able to properly report it is really important, because if we set up a database for people to report these different plants, if they, for example, see a Chinellii or a cristata and report it as a ciliaris, it's going to cause all sorts of issues. So trying to figure out a way that we could set up selection criteria to narrow down exactly what the person has seen is really critical. And it's really, really hard when some of these almost look identical. So part of my training for my two sons during our coronavirus hiatus from actual real school was I took them out into the swamp and we identified these different plants. You can see here Plutanthera ciliaris. This is taken in the green swamp at an area known as the Big Island. If you're ever in this area, there's a tremendous amount of different orchid species. I could probably name 15 or 20 in this area, as well as a lot of different types of pitcher plants and Venus flytraps, which are native to eastern North Carolina. There's quite a few of them out here. Also here, interestingly enough, is Spiranthes etonii, which had not been seen in North Carolina for probably 50 or 60 years, and people thought that it was basically just, it was no longer going to ever be found. And we found a thriving population in the back of this particular savanna. There was probably 100, 150 plants. Just very, very small, very hard to see, and not, unless you're there at the right time and actually staring really hard at the forest floor, you're probably never going to see it. But it turns out that it was actually there and doing quite well. So here's the website that you can go to. If you want to find it, you can Google Go Orchids, or you can actually type it in. It's just goorchids. NorthAmericanOrchidCenter.org, but if you Google Go Orchids, it'll take you there, and you can click on Go to Simple Key. And this key will take you to a page where there's a lot of different selection criteria. For example, what is the habitat, where were you, what was the actual leaf arrangement, describing the different parts of the flower, and then as you narrow down the filter results on the left, the selection of orchids on the right will gradually get smaller. And hopefully, the hope here is that you can narrow it down enough to find a picture of the actual orchid that you're seeing. And then you can click on it. Then you know what you can actually, report that you saw it. And that's one way, and probably the only way, that we're actually going to be able to determine where these species are and how they're doing. Outside of having a handful of people just canvass hundreds of thousands of square miles of forest. We're really just crowdsourcing a lot of searching for these. So I will stop there. This is photo from the Green Swamp, but did want to give a big shout out to the National Fish and Wildlife Foundation for a lot of their work in the longleaf pine savannas, doing a lot of restoration there with prescribed burns. It's critical to do these prescribed burns for their carnivorous plants as well as the orchid species. And then the Smithsonian Environmental Research Center, where they're actually studying a lot of the different the genomes for these different particular fungi that have associated relationships, and trying to figure out how to propagate these native orchid species in captivity. And then the NEAR system, because a lot of these orchids are actually on your property, so if you're out there, thank you for preserving them. And then the Nature Conservancy. So a lot of times when we find these orchids, if they're a threatened, protected, or endangered species, the Nature Conservancy and other nonprofits will come in and buy up the land, and they'll offer fair market value to protect the species, because once you actually find a threatened species on property, you can't really build on it. And the landowners find it's a great deal because they get a decent amount of money for their property. The property also gets protected, the species gets protected, and the truth is a lot of this is swampland that you probably wouldn't end up developing anyway. So it's a win-win in the conservation effort. So I will stop there, and we will open it up to questions.
Sure. This is Kate. Dr. Jacobs, thanks so much. I'm going to start with the first question. Can you hear me okay? So our first question is from Leon, and it's back to the NOAA initiatives that you were speaking about in the first part of your talk. So this one is all NOAA code going to be released to the public? So can you speak about NOAA code and some of the public-private partnerships? So
hopefully going forward, yes, certain versions of all the code will be released. I mean, it's all taxpayer funded, so all of the public has access to all our code. Whether or not it makes sense would be different. There's certain aspects of our code that are hard-coded towards our own hardware, so I mean, the public's welcome to it. I'm not sure what they would do with it, but the ultimate hope is to transition all of our code to platforms that the community has access to, for example, different type of architectures. And so the hope is that we get as much out of it out to the community as possible. So we're one by one, we're putting all the different pieces of model software on GitHub in no particular order, but the hope was to really get the medium range weather forecast out there, the GFS out there first. I think WaveWatch 3 is out there, the UFS, the short range, and maybe the S2S version is out there too. But ultimately, it's all going to be out there.
afternoon, Dr. Jacobs. The second question we have is from David, and he would like to know, can you use AI on a photo to identify the orchid?
Maybe. That would largely depend on the quality of the photograph, but I would imagine yes, provided that the photograph is good. I can tell you where we have used AI is to identify likely sites for these orchids. So one of the things that we've been looking at, we have digital elevation, we have angle of sun, we have how much shortwave sunlight or the radiation can get through the canopy, we have slope of the ground, we have all the different type of pH and soil content. There's a lot of different components. And we can use AI to sift through all these different USGS files and land use files to determine where the most likely places to look for would be. And there was a big project done in Prince William National Forest where they went through every different variable they could think of and then identified where they would expect to find orchids in the forest based on all these different metrics, and then went out and looked. And it turned out that it verified quite well.
Thanks so much. Dr. Jacobs, our next question is from Mark. And Mark asks, what does leadership look like to you for this particular project related to orchids and your citizen science endeavors?
So I would say leadership is probably more of just trying to share my enthusiasm. It's really hard, and this, you could probably apply this to almost anything. You can't just go out there and tell people, hey, you should do this because it's a good thing to do or whatever. But if you're really excited and passionate about it, it's relatively contagious. Other people get interested and excited and share in that. And so it's more of a leading by doing. But without actually sharing this, no one would know that this is being done. So I haven't given a talk on this orchid project since I came to NOAA, and it doesn't mean that the project has been sidelined. It's just something that's really not particularly central to my job at NOAA. But being able to go out and talk about this and actually use my position at NOAA as a way to help get the word out more about this is I think, is one component that's interesting. The other thing is just being able to get other people excited and interested. And some of that is giving talks like this. Some of it is that citizen science component with the Smithsonian where they're actually going into the classroom. And a lot of it is just general awareness. Like I said, you may look at the side of a sidewalk and see a plant and not realize an orchid when it actually is.
Thanks for that, Dr. Jacobs. The next question is from Ken. Do you ever send samples in for genetic identification? And have you personally taken any pictures of previously unidentified species?
So no, well, so to answer this last part of your question, I haven't actually discovered any unidentified species, but I have discovered an unnamed hybrid. The last species I think that was named, that was recently identified was, and recently I mean maybe 15 years ago, was Corallorhiza bentleyi. It was named after Stan Bentley, who was a naturalist in West Virginia. So yeah, if you actually do find a new orchid species and can genetically determine it's a species, write the paper, name it after yourself. For those of you who actually know, my cat Stanley is named after Stan Bentley. The first part of it is, yeah, so it's one thing to talk about the morphological features and claim it's a new species. It's another thing to actually sequence the genetics of it. And my wife is an evolutionary biologist at Duke who does a lot of gene sequencing at a core down there. So this is not actually what she does for her day job, but she's quite familiar with that. But most of the sequencing is actually done with the Environmental Research Center at the Smithsonian.
Interesting. Dr. Jacobs, this question is from Kristen. Kristen asks, what's your favorite orchid, and how did you initially get interested in them?
I have to say maybe either I don't know. I can't really answer that. You got me stumped now. Probably Isotria medialoides, just because it's so hard to find, and there's so little known about it. But how I actually got interested in this, I was standing on the side of the road. I was riding a bike with a friend of mine, and we had stopped, and we were standing on the side of the road, and he was a botany horticulture major, and I just looked down and I saw a pink lady slipper, and I was like, that's a cool plant. And I had actually been growing tropical orchids as a hobby, but I didn't really know anything about native orchids, and he was like, yeah, it's an orchid, and then proceeded to tell me that there was at least 70 species just in North Carolina, and I thought, maybe I should go look for them all. And then the next thing you know, we were rewriting 2 giant textbooks.
Thanks for that, Dr. Jacobs. Jack would like to know if you could speak the name of the hitchhiker variant that you mentioned that's native to Europe, and if you could show that photo again.
Sure, it's Epipactus Helle, and you can see the name down here. It's actually literally pronounced helleboring, as if you were from New Jersey and weren't interested in this plant.
That's a good photo. So Hillary has a question for you, Dr. Jacobs. Hillary is asking about when you were highlighting kind of some of the conservation concerns for native orchid species, and she cites specifically poaching and habitat destruction. And then, obviously orchids, like other plants and wildlife, can also be impacted by extreme weather events. She asked about what role you think reintroduction and specimen salvage will play for the future of our native orchid species. And do you think land managers have a responsibility to intervene to prevent, kind of the loss of orchid populations?
I don't think it's necessarily up to the land managers, to be honest. A lot of times, if these are actually listed as endangered species, it'd basically be against the law. But in general, awareness of it. So like I said earlier, reintroducing these things into the wild is actually a really complex problem, because they're so dependent on other species, whether it's the fungal relationship, and if that fungal relationship is related to a type of tree. For example, I think that maybe the Mediola, there's some discussion about whether it has a relationship with either a birch or an oak, and so you could reintroduce the plant all you want, but unless you reintroduce the fungal host trees and everything else associated with it, it's still going to be tricky. It's not something that can't be done, but the conservation effort needs to go well beyond just the actual orchid itself. The other thing, as far as habitat conservation, that's just the general awareness of what's out there. And this is, this probably goes for any plant or animal species that's highly dependent on its habitat. You wouldn't know to protect it if you didn't know it was there. And so while some plants and animals are pretty obvious, some of these other ones, unless you actually went looking for it, you wouldn't know it was there. So a lot of times, particularly I think in New Hampshire and Vermont and other locations, if you want to get your land zoned for some type of development, you will have to hire a botanist to come in and do surveys for particular endangered plant species. And one of the species they'd be looking for is Isotria medioloides.
Thank you for that. The next question is from Mary, and they'd like to know, in what meaningful ways can NOAA line offices enhance citizen science contributions to our program?
At a really high level, just more involvement and more outreach, and particularly outreach in the K through 12 area. You know, I don't, whether you're going into the classroom or doing camps, it's relatively easy to reach broader people through the internet now than it used to be. But I really think, and this is of course a little bit tricky in the times of COVID, but having actual real field trips where people get to sort of hands-on experience with a lot of these things. It's when you grow up in schools, particularly in urban areas and cities, and you know your exposure to the outdoors might be at the playground, you're not actually getting out into the wilderness and experiencing nature in its natural form, and I think that's probably critical for people, particularly in urban areas. It's really, you got to make an extra effort to get the younger kids really exposed to the natural world, and it's not necessarily easy because you might actually have to travel kind of far.
Dr. Jacobs, this might be related a bit. Lisa asks, she says, the legacy of citizen science runs deep in your family. How do you encourage your young children to be involved and instill that generational legacy and make sure that it passes on to them as well?
I think that appreciation of the natural world is something that is they like because they've grown up. You know, we teach them when we're out in the woods to just make sure you watch where you step. We never pick these plants. It's just being respectful of the environment and sort of teaching them about it. Appreciation of it will come as long as they understand it and are there and a part of it, but it's really hard to appreciate something that you've never necessarily been exposed to. So my goal was just to expose them to as much as I could of the natural world and hope that the appreciation just came along with it.
Pardon the noise, I have a train going by right now. Jack would like to know, have you looked into smartphone apps such as PlantNet for crowdsourcing the orchid ID and location work?
I haven't. I know that others have. There's been a lot of work on this in the last, I would say, 2 to 3 years. But I've sort of been a little bit out of the loop just because I've been so consumed with NOAA things. But I can say there are people working on this and trying to integrate a lot of smartphone apps into the databases, which ironically is almost the same technical challenge that we have in the marine fisheries with a lot of these recreational fishermen apps.
Thanks, Dr. Jacobs. This question is from Diane, and she asks, can you please tell us how you obtain your surface radiation measurement? Is that?
Well, so you could actually, you could do it a couple different ways. You can either you can actually put equipment out there to measure it, or you can use model analysis. So I don't necessarily know how reliable the model analysis is particularly. It'll give you a good shortwave, long wave down and up with respect to how much is attenuated from the moisture in the clouds, but it's not going to properly resolve the information based on canopy cover because a lot of the land use files are pretty old. So, and they don't necessarily really account well for deciduous trees. So obviously your fluxes are going to change if there's more leaves on the trees blocking the sunlight. So the best thing to do for that is actually put instruments out there.
Good afternoon. This one has been sort of touched on before, but Elizabeth would like to know, do you think that instead of creating hard copy books, creating just a digital archive on a website instead would be something akin to moving to the cloud, would put things together faster?
Yep. So that we have done. All of these files are the digital versions of the file. In fact, all the photos. This is the beauty of digital cameras these days, is you don't actually have to scan slides. Everything's digitized. It's archived both in Flickr and Ipernity and a couple other databases. I don't actually necessarily know that the next version of the book will be a printed out hardbound type book. It could very well be digital.
So this is a question from Diane. Do wild orchids grow in western Pennsylvania?
Oh yeah, definitely. I would say off the top of my head probably at least 20 or 30 different species that you could find there. I've never specifically photographed any in western Pennsylvania, but I have photographed quite a few in that area, particularly in central and eastern Ohio. There's actually a unique species of Superpedium that I've only seen in that area.
I believe this is our last question at the moment. Dan would like to know, does Go Orchids Simple Key contain only threatened and endangered species? There are 226 species listed in the Simple Key.
No, I don't necessarily know the order of the different species that are being uploaded there. I don't think that there has been an effort to put the threatened ones up first. I'm only saying no because I know that there's at least a couple I've seen on there that aren't necessarily listed as threatened. But as to the priority of the order of what gets uploaded, I don't know how that actually works.
Dr. Jacobs, I think that was the last question. Thank you so very much for a really fascinating talk. And thanks everyone for joining us today. Our next seminar in the NOAA Environmental Leadership Series will be on Tuesday, November the 10th at 2 p.m. Eastern Time, featuring Vanessa Griffin, Director of the NOAA NESDIS Office of Systems Architecture and Advanced Planning, OSAP. See you all then. Thank you very much. Have a great day, everyone. Bye-bye.